And we will start today's presentation. So I was mentioning, if you are watching us online, we were supposed to be joined today by Dr. Kim Moore. He's having some connection issues. Luckily, we had up on ready to bat Dr. Henry Choong, who will be our presenting today. And I welcome you to this improv Thursday, the 16th edition of RBCM at Home. My name is Kim Goff, and I'm a learning program developer at the Royal BC Museum. I hope you're keeping well and staying optimistic. I'm coming to you from my home on the territories of the Lekwungen-speaking people in Esquimalt. Like many of you, staff of the museum have been asked to work from home during the pandemic. And during the series, we're exploring a bit what that looks like, and we're discovering it looks different for everybody. Some people have got lots of projects they're working on, some people are doing research, some are writing, some are working with their colleagues from afar. So today we have Dr. Henry Choong. Henry is the curator of invertebrate zoology at the museum. And Henry, you were telling folks just a little bit about what it is that you study.
Okay, I study hydrozones. I'm a taxonomist, which means I study a particular group of animal and how they're related. So my specialty is a group called hyrdozoa, and hydrozones include animals that at one stage of their life kind of look like plants. So that's why I suggested to Kim today that if for some reason Ken could not connect, at least I'll be able to show you some animals that look like plants. And they even grow like plants. In some ways, they behave like plants, and maybe that'll, that will also show folks that a lot of the things that we do, even in different fields, are not that different. So.
Okay, excellent. Thanks, Henry.
And I can show people a picture of what hydroids look like in case there are people who have not seen these before.
Yes, please.
Let me just put that up on the screen here. Here we go. So that's kind of a general slide that I have of what hydroids look like. Now, hydroids are, for those of you who don't know, are related to corals and jellyfish. They're cnidarians, which means they have stinging cells, as all cnidarians do, which is why jellyfish can be quite painful if you touch them. And hydroids are a group of cnidarians. And what's interesting about hydroids, about hydrozones, is that hydrozones have a two-stage life cycle, and most of them, many of them, will express this two-stage life cycle in which at one stage they look like what to most folks may appear to be a jellyfish. And another stage of their life, they look very much like plants in that they are fixed to a substrate, and in the picture here you can see on the right, a couple of the pictures show you what some of the hydrozone polyps look like, and these are growing on. These grow on both natural and artificial substrates, which means they grow on, they can grow on rocks, they grow on other animals, they can grow on other hydroids, and they can also grow on artificial substrates, ropes, plastic, styrofoam, almost anything. So and that mode of life has a lot of implications for for what they do in terms of biodiversity and what we need to know about how they live and how they reproduce and so on. And it also influences how we approach their study. And when I say they appear to, they sometimes look like plants and they behave like plants. A lot of the terminology that we use, for example, is similar to what is used in botany. So Ken and I are able to speak about these animals using the same language, although sometimes we apply that in different ways. So I always thought that was interesting in my conversations with him.
You speak the same language.
Yes, more or less,
Yeah.
more or less the same language. I mean, for example, we talk about branching patterns. We talk about roots. We talk about root systems. We talk about even though hydrozoans don't have roots as plants do, they are some hydrozoans are colonial, or many hydrozoans are colonial, and so they will have colonies where they're branching and all the different polyps are working together as one, and so nutrients are distributed through the system, not exactly like plant, but very much like that. And some plants are like hydroids, in that some plants are predators, and hydroids are predators. So there are some similarities. I mean, they were, in terms of how we just by their morphology and their mode of life, how we approach the study of animals. So I always found that quite interesting, and I always enjoy my conversations with Ken. And we've had conversations about how to describe shapes of hydrozone colonies, because a lot of what I do is in describing them is, we find that a lot of the terms that we use, again, are the same as what they have in botany or similar.
Speaking of appearances and telling the difference and observations, there's a question here, and they're asking, how can you tell the difference between a hydroid medusa and a jellyfish?
Okay, that's a very good question. Okay, so you can see in my one slide there, and Kim, I promise I only have one slide today.
Oh.
So and so you see in that picture there, where I have
if you use your cursor, I think we'll see it on the screen. Okay. If you want to show us which one you're talking about.
This one here.
Okay.
Can you see that? Okay.
With the black background. Yeah.
Yeah. So that's a picture of a medusa, and in fact, behind me in my virtual background is also a picture of a hydrozoan medusa here, and you can see. So how do you tell the difference between what we call a true jellyfish and a hydromedusa? Hydromedusa is what we refer to normally when we're referring to hydrozoan jellies, right? So for starters, true jellyfish or scyphozoans are generally much bigger than hydromedusae. Hydromedusae tend to be very small, and you can see, can they see me on the screen right now, or just the slide?
Yes, they see you in the top left corner.
So I'm holding my fingers like that. So hydromedusae tend to be fairly small, although some jellies are small as well, but hydromedusae tend to be in general smaller than jellyfish. And they also have one feature that jellyfish don't have. If you can't really see it, well, I'll just kind of point out where it would be. So this is the bell, and around the bell here, hydrozoans have a muscular structure, or hydromedusae have a muscular structure called the velum. It helps them to contract. It's a muscular structure here, and jellyfish don't have that. So their anatomy is different. So that the one sure way to tell a hydromedusa is the presence of vellum.
Now, what about location? What about where you would find them? Would you find them in the same places?
Sometimes yes, because if you if you're able to go to docks, for example, at the correct time, you'll often see many different kinds of gelatinous organisms floating around out there or swimming around out there. And not all of them are jellies, not all jellyfish, not all of them are scyphozoans or hydrozoans. But you'll see hydrozoan jellies and jellyfish in the same place, yes. Although the hydromedusae may be harder to pick out because they're generally much smaller.
Yeah, okay. Also, I guess I thought of hydroids as always clinging on to something. I guess that's not the case. They can be swimming freely.
No, that is the case. When we refer to hydroids, I should have explained this. So when we refer to hydroids, we're referring to the polyp stage, and the polyp stage is the fixed stage. And that's why they're clinging to various substrates and so on. When we refer to the medusa stage, we generally don't refer to them as hydroids, although there may be medusae that are related to certain hydroid stages. So, and what complicates the picture is that these two stages exist in hydrozoans, but not in all cases. So in other words, some hydrozoans have both the polyp and the medusa stage. Others, in others, we've never seen the hydroid stage, and in others we suspect that they are producing medusae, but we've never seen them, right? So, and how that affected taxonomy was that for the longest time, hydroids and medusae were classified under different systems and parallel systems. So in some cases, the same animals, basically in two different forms, were classified totally separately. And so one of the things that has happened in hydrozoan taxonomy and systematics is that we're putting that system together and linking that system together. And a lot of these stages are not seen out in the field, so to speak, because they're so small, and because it's simply very hard to see that. So some of these stages have only been seen in the laboratory, for example. So there are people who are doing life cycle studies, and they're raising we're raising the medusa so they can see how they change during the lifetimes, and from which polyp they came from, and so on.
And that's part of your role at the museum, I suppose, is documenting that and collecting that. Are there specific or particular challenges to storing them within the museum?
Yes. So because they of the nature of these two life cycles, they have to be preserved in different ways. So when we are preserving hydroids here, there's my cursor pointing that my cursor is pointing to.
The top right picture.
The top right pictures. Generally, let me back up a bit. So it's never it's never as simple as we'd like it to be. So even within hydroids, for example, there are different ways of preserving them. So in the top picture here, you're seeing a hydroid called Simulaclava, and this is what's called an aethicate hydroid. And aethicate hydroids do not have a covering in which the polyp can retreat into. And thecate hydroids down here have a cup, or a theca, which is where their name comes from, in which the polyp can completely retract to, in most cases. There are always exceptions. And so, and so when you preserve them, we usually preserve our specimens in ethanol and 70% or higher ethanol. And for aethicates, they tend to shrink, they tend to shrivel if they're not prepared carefully. And they tend to shrivel more in ethanol than the thecates would, for example. So we're very careful. Now, traditionally, the aethicates up here have been preserved in formalin because that preserves if they're fixed and preserved properly, the formalin preserves the tissue much better. And the same for medusae, because medusae are gelatinous. They tend to dry out in ethanol, and we preserve them traditionally in formalin. So these days we preserve a lot of our specimens in ethanol, both because ethanol is less toxic to work with, and because we can preserve the DNA better in ethanol. But in general, for the morphology or being able to be able to look at their shapes, to be able to look at their structure, we tend to preserve the hydroids in ethanol and the medusae in formalin. Although, and I do a combination of both.
So what are some of the big questions you and other scientists studying hydroids are trying to answer about hydroids?
Well, one of the questions, the big questions that I've worked on recently with my colleagues, many colleagues, that one of the questions is, how do they travel? How do they spread? Because, as you know, being rooted to a surface, it's much harder to spread than if you're free swimming. And so you might think that that the way to spread is through the medusa stage, right? Because they are because they are not attached to any surface. Again, speaking of exceptions here, some medusae have little suckers that enable them to cling to different surfaces. So they're not rooted, but they can cling to different surfaces. So you might think that pelagic stages, or free swimming stages, are more easily spread than the fixed stages. But in terms of long distance transport, and for example, if we're talking about transoceanic transport, now how do you get something like that happening over thousands of miles? You find that it's not the pelagic stage that is significant here, but it's the polyp stage. Why? Because the pelagic stages usually don't live very long, and while they are capable of being dispersed, through in the water, they're not as effective, and they can't survive long distances like the polyps can. And why? Because the polyps are clinging to different substrate. I mentioned artificial substrates earlier, and these artificial substrates, which we are producing now, such as plastics, styrofoam, and so on, these substrates do not degrade very easily. So they persist for a long, long time. And because you have this stable substrate for the polyps to be on, then if a colony or a hydroid or a colony can establish itself on one of these substrates, and the substrates can be carried far afield, then you can have colonies that not only are capable of being transported across long distances, but you have colonies that are able to reproduce along the way and so can be transported. So that's one interesting aspect of hydrozoans that we're studying, and also in terms of their transport. And how they're able to survive such long distances, is hydroids also have this capability, great capability of, when times are hard, and there's a lack of food, or when conditions are because in the open ocean, conditions are very harsh. So under those conditions, hydroids can actually shrivel and shrink to they can actually shrink to what's called a menon or resting stage, and we think that also allows them to survive these long distances.
Henry, we have a question here for you. What is their function in the marine ecosystem, and are there any freshwater hydrozoans?
Okay, yes, they are. There are hydrozoans. One freshwater hydrozoan that many, if not most of you will know, are hydras. So hydras, they're generally presented as typical hydrozoan, but they are far from typical, and they're very, very interesting indeed. So yes, you do have hydras, for example. One example of a freshwater hydrozoan. There are hydroids that live in brackish water, and so on. So we find them across a variety of salinities, but mostly they are mostly marine. And now, what is their function in the environment? Well, hydrozoans are predators, and they like many other predators their size. They will eat fish larvae and little marine creatures and so on. So they act as part of that system. And the other aspect of them is they also form food for some fish will eat hydroids, for example. And the other benthic marine animals will eat hydroids. Some nudibranchs will eat hydroids. In fact, some nudibranchs will associate themselves with certain hydroids. So the hydroids can function also as anchors in the ecosystem. So and what they do is they in turn, so they will be established on a substrate, and then they in turn will act as a substrate for other animals, both living on them, living around them, in some cases living in them. So we see if I can find another. I better not attempt to find another slide. But they're an important component of what are called animal forests, in that they serve the same purpose. And that's a very good question, actually. They serve the same purpose as trees and shrubs and do in the terrestrial system. So they form this whole area where other members of the community are able to live in and get their food and so on. In some cases, the hydroids will serve as protection for other animals. If you sometimes you'll see hydroids covering hermit crabs, the shell of hermit crabs, and what they do is they function there as because they have little stinging cells. They'll serve to deter other animals from bothering the hermit crab, and in turn, what they'll get is any bits and pieces of food that the hermit crab doesn't eat. They'll get some of that, and plus that will also let them have some mobility, because the hermit crab can move around, but hydroids can't.
Wow, it sounds, yeah, such a wide variety that's there. Henry, I'm going to ask you to stop sharing your screen for us.
Yep.
We'll see you large and in life once again.
Okay, I will do that. I will attempt to do that. There we go. Okay.
Great. It has been so great of you to step in for us and to talk about your passion. I guess that's not a really difficult thing to ask a curator to do, because that is sort of the magic of curators. You have deep knowledge and deep passion for what you're studying, and thanks for helping us understand why you are so interested in hydroids and all that they're there to do. I see there's, I think, one more question. Oh, there's a thank you that popped up here. I thought it might be a question that has popped up. I also thank you. That was Wynn, one of our participants who is watching today, who says thanks. We will try to get Dr. Ken Marr rescheduled for a future talk so we can hear more from him in the future. And with that, Henry, I want to again thank you for stepping in and helping us out today, and well, providing the whole content today, so thank you for that. And next week on Tuesday, I hope you'll join us when Holly Hodgson, the manager of volunteer services, is going to be joining us for National Volunteer Week. We'll also have some volunteers here sharing their passions and what they do in the museum that helps to make it all possible for us. So we'll see you on Tuesday. Also, if you have young ones in your family, you might want to check out RBCM at Home Live Kids. This is hosted by our marine museum educator and our staff, and they usually involve hands-on activities. And we're also doing RBCM outside, where we're going out when safe and sharing a little bit of the outdoors, because we're all probably getting a little bit of cabin fever these days. You can find all of those programs on the Royal BC Museum website, and recordings are also made and posted later, so you can share those, or if you want to go back to see them, you can do that as well on the Royal BC Museum's YouTube channel. So thanks for today, and everyone stay well. And while we wait for better times, stay connected in these safe ways, joining online and when you can. So do take care, everyone. And again, thank you, Henry.
Bye.
I'm just going to